Next up, we have Dr. William Lee from Helix. Thank you.
Hi, everyone. Thanks to Twist for inviting me to chat about some of the work that we've been doing at Helix. So I'm going to talk about and touch upon some of the topics that Emily just talked about in terms of rapid customization of the assay that we run at Helix, which we call the Exome Plus. But you could think of it as an enhanced medical exome. So first of all, for those of you who don't know about Helix, I want to give a little bit of introduction just to give some context for why this work was so important to us. At Helix, our mission is really simple, and it's really focused around democratization of genetic data. And we believe that, in the future, everyone should have access to their genetic data, be able to do with it what they want, and be able to learn from it, and have control over it. And so we chose to kind of get at this problem by building a platform for DNA-powered products. And so you can think of this a little bit as kind of genomics as a service, but in a slightly different way, where it's a little bit more like cloud computing, where in the past, maybe you had to buy computers, you had to hire people to administer them, and you have to maintain all that infrastructure so that you could develop your web application. And we're trying to do something analogous for genomics, where if you want to build a product that incorporates DNA data of any kind, we've built a platform that handles the sample collection, the sequencing, the storage, the analysis, and you can really just focus on the software component of that. And so, just to put that in a different perspective, the idea really is that our partners can focus on the pieces that matter the most to them and their customers, which is the direct relationship with the customer in terms of the acquisition and the product experience, and Helix will take care of the rest. And so if you're a company that doesn't have sequencers, that doesn't have bioinformatics people, you can just come to us and integrate with our APIs to be able to access that kind of information. Just for some examples, we have partnerships with, for instance, National Geographic, with an ancestry application. Mayo Clinic has a product called Gene Guide, which is a little bit more of sort of an educational product around how genetics might impact your health. And of course, the backbone of all of this is the lab and the assay that we run in the lab, which I'm going to talk about for the rest of the talk. And so this is a CLIA-certified clinical lab that's located in San Diego, and we do only one thing in that lab, which is run this assay that we call the Exome Plus. And every single exome is signed out, is clinical grade data, and I'm going to talk a little bit more about sort of what comprises that Exome Plus. But a lot of the effort was not just spent in development of the assay, and we did a lot of the early work in collaboration with Illumina, but also in building up processes and quality systems so that we can have a really high quality product and process and know it and have that be documented and traceable. And then also sort of be able to continually improve it, because as you all know, the technology is constantly changing, and if you stay in a certain point of time, you will fall behind. And so I think for this audience, this is probably a little bit more of what you're interested in. And so we call this an Exome Plus. You can think of it a little bit as a medical grade exome in conjunction with content that can allow you to do applications that you would normally think about doing with a genotyping array. And so that means that we've prioritized coverage for a bunch of medically informative genes. I'll get into more details about that later. We have broad coverage for all the other protein coding regions. We've spiked in about 300,000 non-coding regions that comprise what we call sort of a SNP backbone. And of course, this is all part of a clinical test. So Helix launched sort of in production with this assay about what was it, probably towards the end of 2016. And then about sort of a year and a half, a year or so after that, we had gathered enough data, being in the consumer business, the data comes in relatively quickly, to be able to see that, there was room for improvement. And after being in business for a little bit of time, we understood a little bit more about what our partners and customers were interested in. We understood a little bit more about what limitations we had in the current assay. And so around, a little more than a year ago in early 2018, we undertook an effort to really focus on improving this and rolling out kind of what we call Exome plus V2. And so what do we want to get out of it? I mean, the same things everybody wants, right? You want it better, faster, and cheaper, right? And most of the time, I think they say you can really only pick 2 out of those 3. And so, but ideally, the more you can get, the better. And so to us, cheaper really meant less cost per sample, which, as many of you know, probably means, more samples per run, right? You want to be able to fit, you want to do less total sequencing, right? And so Emily talked a little bit about that in terms of, better uniformity. Better for us in our applications meant improved coverage of the regions that our partners and customers cared about. And faster, really meant higher throughput. So being in the consumer business, the volumes can be quite high, and they're quite seasonal, as some of you may know. And so during certain times of the year, you have a lot more samples coming in, and you want to maintain a good turnaround time. And so having good throughput, being able to schedule shifts in a way that you can increase that throughput more easily is pretty important. And so we started feasibility work on this kind of around January 2017, and then things went pretty fast, and this was pushed into production around November of last year. And so just to get into a little bit more detail about the types of things that we looked at. So version 1 of our design was, I think, at the time already pretty novel, where we took an existing exome and we boosted coverage and tried to finish regions with a newer gene model annotation for about 5,000 genes that we considered our priority genes. And in addition to that, we spiked in around 180,000 non-coding SNPs. And we deliberately tried to sequence those 2 at different coverage levels. And so I want to direct your attention to the bottom, where I don't think anyone can see the pointer, but as most of you know, with targeted enrichment applications, hybrid capture-based applications, if you have your exon, which is kind of that rectangle, you get coverage that looks something like this, right? And the trick is that you have more coverage in the middle than you have the edges, but the edges still matter, right? Maybe even a little bit beyond if you care about splice sites. And so you need to sequence enough that you still get good enough coverage on the sides, right? And so your mean coverage or your average coverage needs to be relatively high to deal with that for the coding regions. Because of the SNP content that we had, though, was dramatically different, right? It was literally like a single base that we cared about. And so you actually don't want all that other stuff on the side. And you really want just minimal coverage in the middle. And so because of that, we didn't really want to put the probes in at equal ratios and get, the same amount of reads assigned to like one set of content as the other. And so we really worked on developing this sort of multi-tiered content. And that worked out pretty well. And over time, we were able to see what the variation was like in production. And we decided that we were probably spending, too many of our reads were going to the SNPs. And so part of the V2 design was to, really optimize that even further, where, at the end of the day, the resource that costs money is each of your reads, and you want each read to go somewhere, valuable. And so with V2, we took this to the next level, and we designed 5 tiers of content, where we took sort of those 5,000 genes and we reprioritized all of that, and we came out with a panel that we internally call the Preventative Panel, which is about 600 genes, and most of you would probably know the names if I were to tell you those. But it's really like if you were to think of a clinical panel of the most important genes that you want for disease, purposes, then you would get something that looks something like that list. There was another list of about 3,600 genes that constituted sort of the majority of genes that had been associated with Mendelian disease in humans, and then of course the rest of the exome. And then in addition to that, we spiked in about 18,000 SNPs that you can think of as kind of the GWAS catalog, and a bunch of SNPs on chromosome Y to help with paternal haplogroup matching and inference. And then there were close to 300,000 SNPs that we spiked in, or probes that we spiked in, that we consider to be that we call sort of the backbone. And this is most of what's in sort of the non-coding content from V1, but was greatly expanded and was really focused on improving our ability to perform array-type applications. So imputation, GWASs, polygenic scores, that sort of thing. And the difference between the GWAS SNPs and the backbone SNPs is that with GWAS hits, they may be non-coding, but a lot of times you want to know, you want to have direct evidence for what the genotype in that position is. Whereas for other applications like polygenic scores and GWAS type things, you were probably going to be mostly okay with imputed genotypes. And so the coverage for those two that we targeted was different. And so, just to sort of put this in context a little bit, that means that our V2 design had, I think somewhere close to 400,000 custom probes, in addition to the 300,000 or 400,000 that were part of the core exome. So it was a pretty big panel. I think one of the things that was really satisfying to me as a participant in the R&D process was how we were able to really take a data-driven approach towards the design and optimization of the panel. And so, I mentioned we started work in January. A lot of that was early feasibility, proof of concept work with the formulation. And then we ordered a panel of about 200,000 from Twist that we tested to sort of see how that would perform. And then we went in April with the first full panel design that was very similar to what I described here as the V2 design. We got those probes. We tested them for a few weeks. We really looked at the regions that were not performing well or maybe performing too well. And then we went in with a second design, a month later. We tested those, and then we learned a little bit more from that, and then we went in with the big order for the production pool with our third design, and then started the process of design transfer and productionization of the whole thing. And so all told, in a period of a few months, 6 months or so, we went through 3 iterations of the design where we were able to look at the data in detail, and from that, iterate on the design. And I'll talk about that a little bit more. So one part of that is, like I said before, one of our goals was really to increase coverage of what we can get data on, so that we can support as many products as possible on the platform and for the customers and partners. And so the backbone was comprised of about 290,000 SNPs. A lot of those were based on the work done by the community and sort of commercialized as part of the Infinium global screening array. And so we took some of that work in terms of the common markers that were in there. And the way that we wanted to measure performance for this was, there's a lot of ways to do this, but I think the way that we cared about was really like, essentially, how many SNPs can you impute with a certain degree of confidence. And so we set a cutoff with a genotype probability of 0.95. And then we looked at how many SNPs we could impute. And the other thing that we had a question about was, how little coverage can we really have on these SNPs? And sort of how does the performance of imputation change as you get less and less coverage, right? Because at the end of the day, we don't want to oversequence these things. And so what does it mean to oversequence these? And so what you see here is, on the x-axis is essentially a downsampling experiment, where 1.0 is some amount of yield. And you can see as you go to the right and you have less total yield, you get decreased performance with imputation, right? Because there's less data and less evidence, and so there's less to impute with. But interestingly, the blue line is the V1 of our assay with about 180,000 in the backbone, and then the red is the V2 with close to 300,000. And so you see a steep drop-off at about half coverage. But even at that, with V2, we're kind of right around the performance that we had with V1. And so the performance from this particular measurement was significantly improved, which made us feel good about sort of where we were with this particular application. And so I mentioned I would talk a little bit about some of the other data that we looked at. So as many of you know, high and low GC content regions can be challenging for a number of reasons. And so one of the things that we wanted to test was, now that we have this capability to synthesize so many different pools of probes, why don't we target the regions that we're particularly having problems with that are very high or very low GC, right? So on the left is a bin of 0 to 30% GC content. On the right is the bin of 70 to 100% GC content. And we took the probes that targeted those regions, and we designed new probes, right, that were maybe shifted a little bit from where the original ones were. And we made this a whole separate pool so that we can spike those in at higher and higher concentrations and hopefully boost coverage as a result. That did not work. So, I don't have the data to show here, but surprisingly, if you spike it in too high, you actually start to see a decrease in performance. And so, that was a good experiment because it taught us something. But it wasn't able to help us with this problem, so we had to take different approaches. And the other thing was, I think most of the time when people think about improving uniformity, it's about, how do we take regions that are not performing well and boost them, right? But I think the vice versa is just as important. And you want to take the regions that are, being over-sequenced dramatically all the time and find ways to bring those closer to the rest of the distribution. And so we took an approach where we looked, just as a subset of probes as a test. And so this is about the 18,000 or so that we call the GWAS probes. And we looked at probes that were sort of, I forget, the highest, 5,000 performing or something like that. And we found that most of those tended to be higher in GC content. And so we did a simple experiment where we simply, somewhere in the middle of the probe, we flipped a G to a T. And what you see here in the blue, so on the x-axis is with the previous design, on the y-axis is with this, this base flipping. And you see that, indeed, that helps bring the coverage down for these sort of overperformers, and which improves your overall uniformity. And so that was helpful as well. And so, at the end of the day, the real focus for us was, how do you evaluate the quality of an exome? And for us, it's very similar to the things that Emily was talking about. What we care about is what we call effective yield, right? Which to us is you have, what's relatively static is the total amount of yield that you can get out of a run. And then if you take off the things that are off-bait, you remove duplicates, and then you remove over-sequencing, then you're left with, exactly kind of the yield that you want for the targets that you care about. And so throughout development, we tracked all of these metrics so that we could see how each one changed relative to different conditions. Every week, we ran experiments where we took the exact same libraries and we split them up into up to 4 different enrichment conditions. And so we were able to empirically measure exactly how much each of the conditions that we were testing changed these metrics that we cared about, and at the end of the day affected our effective yield and the callability that we cared about. And so what I'm showing here on the right is kind of the way that I like to think about where we are today compared to where we were before, which is that on the x-axis is total yield, on the y-axis is what we call callability. You can think of this as sort of percentage of your target region that's at 20x or higher. I mean, the absolute number, as many of you know, is pretty subjective, and so it's really the relative numbers that matter. And so obviously as you get more yield, you'll get more and more things that you can cover at the rate that you want, but there is diminishing returns, right? At some point, more sequencing is only going to sort of incrementally get you slightly more of what you really care about. And so there's a sweet spot, right, where if you want to really optimize and put in like just enough sequencing to get the coverage that you want, there's a sweet spot to hit that. And so with V1, this is the curve that we had. And what I'm showing in the green is, an approximate approximation of sort of the 10th to 50th percentile in terms of total yield for the samples that we have in production, right? So the right side of the green box is the median. And of course, what we care about is, at the end of the day, if things drop too far below here, we have to requeue them for more sequencing, right? And you don't want to do that too much. You want to do that as little as possible. And so with the blue curve now, you can see that we can actually shift this entire thing to the left. And we can do less sequencing per sample and obtain, actually better quality. And so, just as a quick summary, we actually succeeded in building an assay that was cheaper, faster, and better. We prioritized those in a particular way, but it turned out that we were able to achieve all of them, which was shocking, but great. And now that it's been in production for a few months, this has really been borne out, in a production environment where, as many of you know, may not be exactly the same as what you see in R&D. And we are seeing higher throughput. We're using less sequencing per sample, and the requeue rates for comparable quality metrics are about the same. So that's it. I wanted to acknowledge people in the Helix team who really contributed to this, in particular, Lorena on the R&D side and Romo on Bioinformatics, who did most of the work. Thank you.